Hello, everyone. My name is Jinzhou Zhang. I'm a grad student from Department of Chemical Engineering, University of Utah. Today, I'm going to talk about long-term tracking of cell lineages with quantitative phase imaging. So why that is important? Why that is important in cancer? It is important for cancer heterogeneity, which it can help us understand the origins of cancer heterogeneity and how this heterogeneity leads to tumor progression. It can also help us understand cancer development with developmental biology, where when we trace the lineages of the cells, it can tell us the development of stem cells, cancer stem cells. It is also play a big role in immunology because we know immunology has application in cancer immunotherapy, where it can help us find the origins of lymphocytes fate decision and can build up model based on that. It can also help us the cell size control model, where we can study the cancer cell size regulation by tracking cell birth size over multiple generations. Here are the challenges in lineage tracing. First, obviously, it is difficult to track cells for days because cells move around and they divide. And when the cell migrate out of the field of view, it's hard to keep tracking of them. Also, we also experience the overlapping of cells location from different lineages. Cells with rapid changing morphology make it challenging to segment and keep track of the cells because it's keep changing its shape. And cells with identical morphology with rapid migration is also hard to trace with. That is because cells becomes identical and it's moves around a lot. It's hard to keep track of the same cell. It can be easily mistaken by its neighbors. So here are some previous approaches. We can definitely use microfluid device to confine the cells in a small space. That perfectly solve the problem of cell migrate out of field of view. It can also help us managing the cell location better. However, it does little with changing morphology and identical morphology problem. We can also trace cells based on its morphology and tracking based on the cell's location. It does not help if this method applied alone. Cells will still move out of field of view, but it can help us separate the cells from different lineages and solve the overlapping problem. Finally, we can label the cell with fluorescent dyes for segmenting. This really helps with separate cells with overlap locations. It can also help with segmenting cells with rapid changing morphology because each cell is identified, can be clearly identified from its fluorescent signal and the morphology doesn't really matter. However, if the cells still having identical morphology and the rapid migration, this labeling method doesn't help much. Our approach is based on quantitative phase imaging plus microwells. With this method, obviously, with the help of microwells, it can help us confine the cells inside the field of view. And our microwells is made of a normal material called MY133, which we will discuss later. It also helps with the overlapping problem from different lineages because we can seeding one cell inside each microwell. This gives us much better management of the location of different lineages. Also, with the help of QPI, our segment is based on cell mass. And our tracking is also based on cell mass plus location. This really helps us to address the problem with cell with rapid change morphology and cell with identical morphology but moves really fast. Additionally, it is common to see adherent cells with rapid changing morphology and non-adherent type of cell with identical morphology and with rapid migration. Our method can both address the problems with changing morphology and identical morphology with rapid migration. The most important part of our approach is that QPI is a label-free method. What that means is it doesn't require any fluorescent dyes to label the cells, which for the immoral cells, the cell lines that can be really easy to be marked with fluorescent and dyes, but for primary cells, it's going to be hard to labeling with fluorescence or other kind of dyes. Our method also include the quantitative phase imaging, as known as QPI. QPI measures phase shift of light passing through sample, which is caused by the refractive index difference between the sample and its surrounding media, showing the image on the left here, where the phase shift of light can be measured. By knowing the relationship between the phase shift and the cell mass, we can calculate the total mass of a cell, showing this image here, where different color represents different phase shift. And we know the relationship between the phase shift and the cell mass. This map also represents the cell mass distribution. And by sum up the entire area, we can know the cell mass inside this area. Now let's talk about our microwell fabrication. Our microwell is fabricated using PDMS using MY133. It starts with SU8 micropillars on a silicon wafer. Then we fabricated a PDM stamp by molding that silicon wafer several times. This allows us to press the PDM stamp on a IBD dish bottom, and the MY133 can be capillary filling inside the gap between the stamp and the IBD dish. The reason we use capillary filling is because it ensures the microwells will be through holes. The reason we use MY33 is because it's a special polymer with low refractive index of 1.33, which is close to water. The reason why I use the polymer with low refractive index is really to avoid the artifacts when we're imaging with QPI, where the phase shift becomes too large and the algorithm has trouble in calculating the real cell mass distribution. After capillary filling our stamps, we left the MY133 to sit overnight to homogenize the different components, ingredients inside MY33, because the ingredients in MY33 tend to separate during the capillary filling. The next day, we cured our setup with UV light, and the PDMS stamp is removed. This left us with the final device of microwells. The image below this picture shows our design factor of the size of microwells. We use 3 different cell types. MCF7 is a common cancer cell line, which is a breast cancer, and it is an adherent cell with rapid change morphology. And 70Z3 is inadherent, is a non-adherent cell line. And the primary B cells is really similar to 70Z3, but it is a primary cell, not a cell line. We designed our height just be high enough to prevent the escaping of cells from the macrowells. And the width is obviously based on the cell size. And we build our macrowell wall thick enough to ensure the strong structure of our macrowells. Here's our setup of the imaging. We made a huge array of macrowells, and the red square here shows the field of view of our camera. And with this setup, we can imaging multiple locations for each cell line for each sample. And each experiment can give us approximately 40 locations if we image 9 macrowells inside the field of view. This gives us hundreds of, possibly hundreds of cell lineages. And with smaller size macrowell design, for example, the macrowells for 70Z3, we have a much smaller macrowells, which means we can fit 36 macrowells inside the field of view. This gives us even more cell lineages coming out of the experiment. Here's how we do the image processing. We first generate an empty macrowell image from the first frame of each location. We take an average of these frames, which gives us a perfect image of empty macrowells. Next step, we overlay our empty macrowells on each images we took, and we subtract away the macrowells, which left us with cells and the uneven background. The final step is to do the background correction, where each cell are segmented out, and we fit a polynomial function to the background and subtract the background away, which left us a flattened background with only cells in the image. And here's our automated mass tracking for 70Z3 over 5 generations. We segment out the cells based on the cell mass and using the algorithm of watershed to separate the cells, and we track the location of each individual cell based on their mass change and their location. The end result looks like this, where it shows the mass growth profile for each cell. And we did a similar thing with MCF7. It is an adherent breast cancer cell with rapid change morphology. And we can also generate the mass profiles over time, similar to the previous 70Z3 cell lines. We can apply the same data processing method even to the primary B cells, where in this case, we are imaging the primary B cells from a mouse, which is subtract from the spleen based on the known particle. We can generate the mass growth profile of the proliferation of primary B cells. And then from the mass profile, we can assign 2 daughter cells to each parent cell. After that, we can apply the tree traverse algorithm, which generate the linear tree from the mass data. Here's the summary of this project. QPI provides a label-free tracking method and use mass as an additional tracking parameter to improve tracking accuracy. Our method can successfully track the lineages of adherent cells, non-adherent cells, and the primary cells. So for the future work, we are interested in studying the heterogeneity and for the future work, we are interested in studying the heritability of the cell mass difference on both sides of the lineage tree and study how that, if there are any mechanism to correct the cell size over the generation. And I really want to thank you, the National Institute of Health, and which gave us the funding of this project. I also want to thank you. I also want to thank the Utah NanoFab, which provide the facility for us to make the microwells, and also Hoffman Lab, which is our collaborator, provides the protocol of primary B cell separation and some insights of cell lineage tracing. Here is the author of this project, which is me and one of our undergrad student researcher and my supervisor, Thomas A. Sango. Okay. Thank you all very much.